Hey everyone, so this video is on antibodies. Again, one of our immune system videos. We've touched a little bit on antibodies in our past few lessons, but this is a more specific video on how they actually work. So our intention is to understand the role of antibodies, to know which cells produce antibodies, and to understand the different types of antibodies that our body has. So what are they? So antibodies, and they're sometimes called immunoglobulins, very, very long, complex word, are just protective proteins that our body produces in our B plasma cells, and we produce them in response to a specific antigen. And we use them to either target or neutralize pathogens that exist inside the body. So the cells that produce them are our B plasma cells, and they have really, really massive rough ERs, and this allows them to produce a large number of proteins, and those are the antibodies. Now, one B cell can produce thousands of antibodies per second. So here you can see B plasma cell producing those antibodies, and they're targeting a particular pathogen. So antibody structure is the same for all antibodies. They're made of 4 polypeptide chains. We've got 2 light chains and 2 heavy chains, and they sort of combine together to form a Y-shaped molecule. So you often see them drawn as Ys in most of your exam questions and textbooks. So we've got a light chain and a heavy chain, and then we've also got a constant region. Constant just means it doesn't change amongst different antibodies. And we've got a variable region, and this is the section that makes the antibody specific. So this is different for all the different types of antibodies, depending on what it's trying to target. So how they work? Well, that variable region will attach to a specific antigen, and each variable region will only attach to one specific antigen. So here you can see in this diagram on the right, we've got this antibody, and its variable region will particularly attach to this yellow antigen. It doesn't fit the shapes of the other antigens, and therefore it's very specific to this yellow antigen. And you can see in the GIF down the bottom, here you've got 3 different antigens, sometimes they're called epitopes, and you can see that this first one binds because the shape of the variable region matches the blue triangle. It doesn't match the square or the semicircle. So they work by attaching to antigens in our body, and then they do a wide range of things after that. So there are 3 main types we're going to talk about of antibodies. We've got opsonins, agglutinins, and antitoxins. They're just different types of antibodies, and they all do a slightly different role, but primarily they're all involved in targeting a pathogen inside our body. You can see here, we've got a macrophage, for example, engulfing a cancer cell that's covered in antibodies. So the first one we'll talk about is opsonins. These basically just bind to the pathogen, and they target them for destruction by phagocytes such as macrophages. So it makes it easier for a macrophage to identify these cells, and it makes it far easier and more efficient to remove these cells. So you can see here's our pathogen, here's our antibody acting as an opsonin, and it's just going to tag that, and then the macrophage identifies that tag and will engulf it and start to break it down. And so opsonins are really, really handy at just allowing more efficient phagocytosis. The second ones are agglutinins, and what these ones do is they actually clump antigens together, or they clump pathogens or even different cells together, and that again allows phagocytes to remove them much quicker. So almost like glue, it will clump 4 or 5 or 6 different pathogens together, or particular cells, and a macrophage can then come and engulf them all in one go rather than individually finding them all. So again, it's just for more efficient phagocytosis. And the last one are antitoxins, or sometimes they're called neutralizers. And basically they just counter the toxic effect of the pathogen. So they block what that pathogen is trying to do. And it sort of masks the impact of the pathogen. So it's no longer having as detrimental effect. And again, it does also make it a little bit easier to start to phagocytose some of these pathogens. So for example, if a bacteria has a particular part of its membrane that's quite toxic to our cells, these antitoxins might bind to that particular section and block its action, much like an inhibitor does with an enzyme. So in review, antibodies are immune proteins produced by B plasma cells. They have that really specific Y-shaped structure, and we know it's got that variable and constant region, but it's the variable region that makes that antibody specific to a pathogen. And the different types of antibodies we discussed aid in phagocytosis and reduce the impact of the pathogen overall. So I hope this video helped. Thanks.